In order to build fatty acid chains, we have to begin with acetyl coenzyme A molecules. Because ultimately, when we're synthesizing fatty acid chains, we're combining many acetyl coenzyme A molecules together. Now, where do we get our supply of acetyl coenzyme A molecules? Well, we obtain it in the matrix of the mitochondria via processes such as the beta oxidation of fatty acids. We also generate acetyl coenzyme A molecules via the decarboxylation of pyruvate, the catabolism of certain amino acids, as well as the breakdown of ketone bodies. But ultimately, this acetyl coenzyme A molecule is found in the matrix of the mitochondria. And that poses a problem, because fatty acid synthesis takes place in the cytoplasm of the cell, but these acetyl coenzyme A molecules are located in the matrix of the mitochondria. So we have to be able to move these acetyl coenzyme A molecules from the matrix into the cytoplasm of the cell to actually initiate the process of fatty acid synthesis. Now, there's a problem with moving acetyl coenzyme A molecules across the membrane of the mitochondria, and the problem is it's water soluble. So if we look at the acetyl coenzyme A molecule, the coenzyme A component of the acetyl coenzyme A molecule prevents that acetyl group from actually moving across the inner mitochondrial membrane. And so to ultimately allow the movement of that acetyl coenzyme A molecule across, we have to remove that coenzyme A. And so what happens is we transfer this acetyl group onto oxaloacetate in the process that is catalyzed by citrate synthase to form the citrate molecule. And now we can move that citrate across the inner and then the outer membrane of the mitochondria. And so this, if we recall, is simply the step number 1 in the citric acid cycle. Now, before we move on to the next steps of this process, actually moving it across the membrane of the mitochondria and then seeing what happens inside the cytoplasm, let's discuss which conditions actually lead to the synthesis of fatty acids. So what conditions do we have to have in the matrix of the mitochondria to actually promote the process of fatty acid synthesis? So in the matrix of the mitochondria, by the way, this is our matrix, our cytoplasm, the inner and the outer membrane of the mitochondria. So we know that in the matrix, we have the citric acid cycle and electron transport chain basically generating ATP molecules. And so when we have high levels of ATP inside the matrix of the mitochondria, we don't want to actually produce any more ATP molecules. We want to stop the process of ATP synthesis. And so what happens is, when there are high levels of ATP in the matrix of the mitochondria, that ATP acts as an allosteric inhibitor of one of the enzymes of the citric acid cycle. So which enzyme? Isocitrate dehydrogenase. Isocitrate dehydrogenase basically transforms isocitrate into alpha-ketoglutarate. And when we have high levels of ATP, it blocks the activity of this enzyme, and that leads to a buildup of isocitrate in the matrix of the mitochondria. Now, isocitrate, if we recall back to the citric acid cycle, can be interconverted into citrate. So it basically converts back and forth. But if we have high levels of isocitrate, we're also going to see an accumulation of citrate molecules. And this will promote fatty acid synthesis. So high levels of ATP and high levels of citrate molecules basically stimulates the process of fatty acid synthesis. And that makes sense because we need ATP and we need citrate to actually begin the process of fatty acid synthesis. So once again, what actually promotes, what stimulates fatty acid synthesis? Well, when the level of ATP in the matrix is high, this means that we no longer need to actually synthesize any more ATP, and therefore the ATP will create a negative feedback loop that will inhibit isocitrate dehydrogenase, and this causes a buildup of isocitrate, which in turn causes a buildup of citrate molecules. And once we actually form citrate, which involves transferring the acetyl group from acetyl coenzyme A onto oxaloacetate, then that citrate can actually move across the inner and the outer membrane of the mitochondria and into the cytoplasm of that cell. So in step 3, we have citrate is then transported across the membrane and into the cytoplasm of that cell. Now, the citrate itself is not actually used in the fatty acid synthesis process. We have to actually obtain that acetyl coenzyme A back. And so what happens is we have a process in which we take that citrate and we form back that oxaloacetate that we began with, and we also generate an acetyl coenzyme A molecule. And this is carried out by the enzyme we call ATP citrate lyase. So in order to regenerate the acetyl coenzyme A in the cytoplasm, the enzyme ATP citrate lyase uses an ATP and a coenzyme to reform the acetyl coenzyme A and that oxaloacetate as the byproduct. So this is the reaction shown here. So we have the citrate that is now on the cytoplasmic side. We have a coenzyme A that is different than the coenzyme A that we had here, because remember, in this process, the coenzyme A was essentially kicked off. We combine these two by hydrolyzing ATP and we form an acetyl coenzyme A and the oxaloacetate. So ultimately, what actually moved across the membranes of the mitochondria are the oxaloacetate and the acetyl group that was attached onto oxaloacetate. And it's the acetyl group that will ultimately be used to synthesize those fatty acids. So it's this acetyl coenzyme A that will now go on to help synthesize fatty acid molecules, as we'll discuss in the next several lectures. Now, what about this oxaloacetate? What is the fate of this oxaloacetate? Well, now we actually have to transform the oxaloacetate into a molecule that can move across the membrane back into the matrix of the mitochondria so that we can recycle and reuse that oxaloacetate. The problem is, oxaloacetate cannot simply diffuse across the membrane of the mitochondria, and we have to transform that oxaloacetate in a two-step process into pyruvate. So let's see how this actually takes place. So we have oxaloacetate. We basically reduce oxaloacetate by using the reduction power of NADH to form a malate. And this is step number 5. So the oxaloacetate cannot cross the mitochondrial membrane. Therefore, to return back to the matrix, it is first transformed into malate by malate dehydrogenase. And this requires NADH. And so we produce NAD plus in this process. Now, once we form malate, an important process takes place that allows us not only to form the pyruvate that can now move across the matrix, across the membrane of the mitochondria, but we also generate NADPH. And this is important, as we'll see in just a moment, because this is the NADPH that we're going to use in the fatty acid synthesis process. So next, the malate undergoes an oxidative decarboxylation step that is catalyzed by the enzyme NADP+ linked malate enzyme, also known as Malic enzyme. And this reaction is important because not only does it give us a way to actually move that molecule across the membrane of the mitochondria, it also generates that NADPH that will be used in fatty acid synthesis. So malate, in the presence of NADP+, and this enzyme NADP+ linked malate enzyme, is transformed into pyruvate. We essentially release the carbon dioxide and we form the NADPH. And it's this NADPH that will be used by fatty acid synthesis. In fact, this is the major pathway that produces the highest number of NADPH molecules that will be used by the fatty acid synthesis process. The remaining NADPHs will be produced via the pentose phosphate pathway. Now, once we form pyruvate, it moves across the membranes of the mitochondria into the matrix, and inside the matrix, we use pyruvate carboxylase to transform the pyruvate into oxaloacetate. So the pyruvate can now move across the membrane into the matrix, where it is transformed into oxaloacetate. So this is the process by which we ultimately move that acetyl group from the matrix of the mitochondria into the cytoplasm, so that now we can use that acetyl group to actually elongate and form those fatty acid chains. And just like I said in the beginning, what stimulates or promotes the process of fatty acid synthesis are high levels of ATP and citrate in the matrix of the mitochondria.